Thank you, and I'd like to introduce our next speaker. Roger Deutsch is the CEO and founder of Cell Science Systems and Privy Medica Group LLC, a nutrition telehealth consulting firm. He is also the CEO of Cell Science Systems GmbH Potsdam, Germany. Roger is one of the modern day pioneers of food and chemical sensitivity testing with involvement in this field since 1986. He has been responsible for the development of the ALCAT test, and he is the co-founder of Your Hidden Food Allergies Are Making You Fat, and has written numerous articles and given many lectures on this topic of food sensitivity, inflammation, and aging on all continents. Once Roger is done, we have speaker Amy Bizarka, which will be joining Roger. And I'd like to introduce her now. Amy received her Bachelor of Science degree in Nutrition and Medical Dietetics from the Pennsylvania State University in 1983. She is also the creator and director of Privy Medica Nutrition Services, and Amy oversees the expertise of registered and licensed dietitians, nutritionists, clinical nutritionists, personal trainers, professional chefs, and health experts committed to achieving the best possible outcome for each and every patient. We are very pleased that both of you could join us today, and I'll go ahead and hand it over to you, Roger, to begin your lecture.
Thank you very much, Tiffany. So I'll just quickly introduce our company. Some people may not know. People typically refer to us as the ALCAT Lab, but the official name of the company is Cell Science Systems. So we are CLIA Lab, and we're also an FDA device manufacturer. And what we manufacture is the instrumentation for the ALCAT test as well as the reagents. And we're the sole provider and manufacturer of ALCAT testing technology. And we do provide these systems for laboratories outside the United States. And Cell Science runs a number of clinical lab tests aimed at prevention and anti-aging. They include micronutrient testing, which is also a cellular response-based test, where we're looking at lymphoproliferation stimulated by a mitogen in the presence or absence of other nutrients. We do telomere testing. We do some other molecular biology for methylation cycling and a lot of gut health investigations. We have a SICA panel, but our flagship is the ALCAT test, and we want to talk about that today, especially in the context of 4 separate research studies that are fairly recent that tie together and are relevant to today's environment with the current pandemic. So the ALCAT test is a whole blood test. Oh, let me start at the beginning. This is what we want to cover. We want to understand the soon-to-be published research from Georgetown University in collaboration with University of Leipzig, looking at the ALCAT as a new test for non-IgE-mediated food allergy, which we also refer to as food sensitivity. We'll explain how the innate immune system is involved in delayed type food reactions and how it's really the specific branch of the immune system that's involved in classical allergy, because there's quite a difference. The ALCAT test is not a test for classical allergy, it's for delayed type allergies. We'll look at how the ALCAT test predicts items, dietary items and environmental items that activate innate immunity that can lead to a number of different pathologies, including autoimmunity. We'll look at how the innate immune system activation through food triggers is determined by ALCAT can be involved in adverse COVID outcomes, and whether or not we might be able to use this technology to predict people who will have a negative reaction to a vaccination, be it a COVID vaccination or otherwise. And lastly, Amy will join us afterwards and explain how the test results are interpreted and how you can integrate this into your practice. So the key here to understand is that the immune system needs to be in balance. I was very happy to see this years ago. I had a subscription to Science, and on this front cover in January of 13 is a lymphocyte with a yin-yang sign superimposed on it. And of course, if you're not completely familiar with that Taoist concept, all of nature is basically expressed through the interaction and permutations and alterations and alternations of the two basic principles. One is more aggressive and masculine and hot, and the other, which is yang and the yin, is more nurturing, feminine, and cold. Now, the immune system we typically think of as being that which causes inflammation, which of course it does. But the other part of the system is aimed at resolving inflammation and nurturing and bringing immune balance and tissue restoration back after there's been an inflammatory response. And they're both very important. If we don't have, if we don't mount a strong enough response to a threat or a pathogen or a danger molecule or a toxin, then it can overcome us. If we overexpress those yin yang qualities, then we can have chronic inflammatory processes, excuse me, that lead to tissue degradation, aging, and autoimmunity, and can progress towards cancer. The ALCAT test, which again is our flagship test, is a whole blood cellular test. It's not a serological test. It is not an antibody. We're looking for a biological response of the peripheral blood leukocytes ex vivo, while the cells are still alive, to a challenge with foods and various other substances, which in our menu will include medicinal herbs, molds, certain pharmaceuticals, and other substances. What's unique about the ALCAT test, and I think this is truly unique, is that the results do correlate with actually eating the food as determined under experimental conditions by correlating with what is the gold standard, a double-blinded, placebo-controlled oral challenge. Not just whether by changing diet the patient feels better. They probably do. There can be, by elimination of common reacting foods, a lot of improvement. There can be a bit of a placebo effect, but the only way to really assess a food sensitivity test is to compare it against a known standard. And the known standard is eating the food, but it should be done under rigorous conditions, double blind, so conditioning is not a factor, so we can control for the placebo effect. And these studies have been carried out on the ALCAT test over the years in different institutions and universities. We're always very liberal, even from the very beginning of our, when we launched this test back in the mid to late 80s, we gave it to university investigators who at that time were really aware that food sensitivity was a major factor in pathology and were searching for a test. We gave it to them, we let them study it, and the results came back that there is a very, very high correlation between what we see in the test well in terms of how the blood cells respond to a challenge and what happens when the person eats the food or is exposed to the food additive under these conditions. Naturally, that improves a number of different inflammatory conditions, notably migraine, notably GI health, notably arthritis and other autoimmune diseases, CNS conditions, and so forth. It also, as we'll see, and this is kind of the central theme of some of the recent studies, is that it also identifies foods that are associated with the release of DNA into the circulation and a process of neutrophil or white blood cell, granulocytic blood cell response to pathogens or danger signals called NETosis. And I'm going to focus on that a little bit because that's kind of the interesting theme, I think, that runs through some of the recent research. And just briefly, and this will just mention, but can be referred to later on, is a meta-analysis of these studies looking at specific outcomes. So what happens when the innate immune system is activated? And by innate immune system, we're talking about the front line, natural immunity, which doesn't require prior sensitization like the specific immune system. Be it a danger signal or a PAMP, a pathogenic molecular pattern, or a DAMP, a danger-associated molecular pattern. Sometimes innate immune cells get activated. They cause the release of toxic mediators that are stored in granules from within, and also generate free radicals, which can spew out, along with contents that are inside the cell, including the formation of neutrophil extracellular traps, which we'll explain, which contain nuclear components, DNA, histones, toxic mediators, membrane components. And when these go out, they are intended to, just gonna jump ahead for a second. They're intended to trap pathogens. This is an activated neutrophil, which has gone into the process of NATOsis. And you can see, again, it stands for neutrophil extracellular traps. This particular photograph sort of set off the interest in research in the field of NATOsis in this particular pathway that the innate immune system uses. In this case, obviously to entrap, ensnare, and apply to the pathogen toxic components, which include DNA and modified histones, as well as things like myeloperoxidase and elastase, which hopefully will kill the pathogen. So after jumping ahead there, I want to come back and fill in the gap. How does this work? What we do with this test is, as I mentioned, it's a whole blood test. So we take the whole blood into the laboratory, we get it overnight, and we dilute it in a buffered saline, and then pipette it or dispense it into a well that contains either a test reagent, could be a food or a chemical, a food additive, for example. And then also some wells serve as controls. And this thing goes through an analyzer, which will count in size the blood particles in that aliquot. We're looking at the white blood cells. So just before analysis, we have a method for eliminating the red blood cells so they do not interfere. And from this, we generate a histogram for each sample. So the control samples are average, and they form a baseline. If you can see it on your screen, that would be depicted by this histogram in the broken line. And then the test histogram for each individual substance is then plotted against the control. And any divergence can be seen. So in this case, we see that the test curve is lower than the baseline curve. And that's because cells have degranulated and they have essentially disappeared. And they may be in the process of undergoing NATOsis, but certainly the membrane has broken down and they are releasing chemicals and free radicals into the surrounding tissue. And that's what it would look like. Sometimes the cells don't fully degranulate. They do swell up. They attempt perhaps to phagocytize a threatening pathogen, and they swell up, and we see the test curve shifting to a higher size range to the right. And of course, if there's no reaction, which we hope to see, that's not true. We hope to find out reactions because those are the things we need to focus on. But when there's no reaction, the curves line up together, and that's what it would look like under the microscope. This graphic illustrates what's happening in the machine where the rubber hits the road. The cells are passing through a sensing zone, which where there's an electromagnetic field created by a cathode and an anode. And they're suspended now in a buffer and electrolytic solution. And these are their cells, and they're exposed to a test substance, and they change in size. And that change in size is sensed by our circuitry. And from that, we're able to generate the size distribution curve that I mentioned. Again, for the controls, which would look like this, and then for each individual test curve. This would be an example of a positive reaction. There's a shift to the right. Some swells have gotten larger, and many of them have degranulated and released their inner contents. Clinically, and this is the first of the 4 studies that I'd like to introduce, we were approached by people at Yale School of Medicine a few years ago who had performed an NIH-funded study surveying the market concerning use of non-conventional medical tests. And this group found out that there was a great preference for looking at food sensitivities using this cellular approach, specifically the ALCAT test. And because one of the principal investigators was a gastroenterologist at Yale, he wanted to look at irritable bowel syndrome. And the study that they designed was a double-blinded, placebo-controlled, randomized controlled trial involving 59 subjects, basically taking baseline measurements of symptoms and looking at the outcomes over 4 weeks and 8 weeks. And those outcomes are depicted here. This is IBS symptoms, global improvement score, and symptom severity score. And this is the treatment group. These are the people who had the ALCAT test diet. And after 4 weeks, this is the change in symptoms, very pronounced. Now, the placebo group also had, particularly after 4 weeks, but then it died down, a benefit. And of course, there's a strong benefit of placebo in IBS, but essentially the difference here was essentially twofold. So the p-value was kind of off the charts that this had to be something that was due to the treatment and not a placebo. So they found that there was no evidence of the placebo effect or the study not having validity because of the way in which the foods were eliminated. Sometimes these kinds of studies are not so well designed because it's very important when you give a placebo diet which foods you're taking out. So sometimes when someone wants to stimulate a good result, they take out dairy and grains, and just that alone randomly produces some effect. In this case, they decided to test everybody with the ALCAT and maintain the rigor and complexity of the controlled diet with that of the therapeutic diet. And the way they did that was this: if they found that a person tested positive to 1 meat, 2 vegetables, 1 nut, and 1 dairy, they would remove that. Again, it was double-blinded. They remove that from the controlled diet, but not the same 2 meats, 1 dairy, 1 nut, 1 vegetable, but a different one, so that the rigor and complexity of the therapeutic diet and the controlled diet were identical. Then they decided at the outset of the study, they banked serum from everyone and then retrospectively looked at changes in serum using a proteinomic screen and found that particularly amongst those, and it was about 12 out of the group that had the therapeutic diet who had the stronger responses, they looked at what changed chemically in their blood. And there were many things that exceeded a 0.05 change. A lot of inflammatory markers reduced, but particularly was neutrophil elastase, that of course is known to be damaging to tissues. Of course, it's aimed at neutralizing pathogens, but it has side effects. And it also will degrade muscle tissue. So that was worth noting. This group had also previously worked studying the role of DNA as a danger-associated molecular pattern in a number of disease states, some of which involve the type of cytokines that have been discussed today, tumor necrosis factor alpha and interleukin beta. And these things are also been shown to be relevant to autoimmune diseases as represented by lupus and ischemic reperfusion injury and NASH, fatty liver. So this led the same team to want to investigate a little bit more deeply because, as you see, with the ALCAT test, we're measuring gross changes in the cell's population. But the question they pose, being the researchers that they are, is what's actually happening inside? And they published their findings, which were showing three basic things that I think are notable. But the one I want to focus on right now is, of course, that following a positive food challenge, positive by ALCAT test, they determined that there was a greater amount of cell-free DNA in the supernatant of that reaction than there was in a negative ALCAT result and or control. So a positive ALCAT reaction would via, and this is another thing they studied, activate this kinase, protein kinase C, which would lead to the result of the release of DNA either from the mitochondria or from the nuclear. Now that's something to keep in mind because they can be different. They can have different effects. Negative response did not do that. And when they used inhibitors for kinases, they found that using the inhibitor for protein kinase C would inhibit these reactions, leading them to conclude protein kinase C is at least a major pathway here, as well as nuclear factor kappa B. And I mention that because when we do intermittent fasting, we're downregulating another protein kinase A, which is related. So it's no surprise that we're dampening down inflammation and bringing about physiological changes that we see that are consistent with healthier metabolism. So I want to focus in now a bit about the NETs. So they've been associated with the following broad categories of conditions: autoimmunity, cardio, cancer metastases, COVID fatality, fatty liver, pancreatitis. And if you look now at PubMed, this is the result of a search that I did a little over a week ago. And these are literally verbatim the search terms that I used. So NETs, again, neutrophil extracellular traps and inflammation, were 1,030 results. COVID-19 complications, 33. COVID-19 fatality, 8 results. Thrombosis, 32. Autoimmunity, 408. Cardiovascular disease, 579. Cancer, over 3,000. Aging, 169. Dementia, 54 results, and continues. Respiratory disease, 529. Treatment of NETs using enzymes that degrade DNA are used right now for some respiratory diseases successfully. So it's beginning to go into the clinic. Pneumonia, 192 results. Probably there's some overlap here. Organ failure, I'm sure there's some overlap with COVID. 47 results. Obesity, 53. Eye disease, liver disease, cystic fibrosis, and cystic fibrosis is being treated right now with, again, DNases. So I just want to cover quickly some of in the interest of time here, the high points. We find that when there's cell-free DNA, this can activate certain receptors on macrophages, cause the upregulation of inflammatory cytokines and the interferons that are associated with autoimmunity. And this has been clearly demonstrated in lupus and arthritis particularly, but a number of other autoimmune diseases. The leukocyte activation test, when it was positive, gave a higher level of DNA content in the supernatant activated by protein kinase C. There was also upregulation of inflammatory marker on the cell surface of eosinophiles most of the time, which is another granulocyte subpopulation that is associated with allergy, getting a lot more attention right now than it has over previous years. And the conclusion is that this test is able to identify foods which cause the release of DNA and activation of peripheral blood immune cells in a PKC-dependent manner. and it suggests that these inflammatory markers will activate symptoms that can range from IBS to other conditions. IBS is just a functional disorder, but it's not very comfortable. This gave rise to an investigation which is just going into print next month. And I'll give you a bit of background. Some of you may know Professor Joe Belanti from Georgetown because he's lectured at the A4M on a few occasions at the fellowships on immunology. He's been working with immunology. He's a professor of immunology and microbiology at Georgetown. And his group had found that certain DNA motifs that were not methylated were more inflammatory than were those that were methylated. And essentially the distinction is this: when you have nuclear DNA, which of course is the bulk of that, for the most part it's methylated because differentiated cells don't need so many genes. So they're mostly switched off. And when they're released, they are not as inflammatory as are fragments of DNA that originate from the mitochondria. And most likely, because mitochondrial genes being much fewer in number, 47, they are, for the most part, not methylated, certainly not in the same sites as the nuclear DNA is. And they resemble much more closely bacterial DNA. So it's not surprising that they would, in turn, be more pro-inflammatory. So what Belanti's group wanted to do in collaboration with our company in Germany and people at University of Leipzig was to distinguish whether or not the DNA that was released into the supernatant following an LCAT positive food challenge was of mitochondrial or nuclear origin. And you do this by using primers and probes that are specific to their nuclear DNA motifs or specific to mitochondrial DNA motifs. So essentially, this is the concept, is to try and determine whether or not the DNA that was released was from either or both of these sources. And of course, we use the Coulter method that we talked about, which has these advantages. It's simpler. It's a very direct. It doesn't require a label like a serological test or certain others, cellular tests. It's just, here's the cells in the whole blood, diluted in a physiological solution, and there's a challenge. And do the cells change their size and number, or do they not? And so it's a very direct and method that represents a final common pathway. And one thing they note is that although there's a number of different factors, danger molecules, so to speak, that can activate the cells in this way, they can also be pharmacologically induced. So we're looking at an innate immune cell reaction, which causes this sequelae, chronic inflammatory conditions that can lead to autoimmunity and so forth. But it can be induced by a pharmacological factor. Meaning, for example, you have in every food, every plant, every fruit, every vegetable, natural toxins that protect them from plants. And if it's been part of our genetic heritage, we've evolved the enzymes to degrade those toxins. If it's something new, and in modern times it often is, we may not be able to fully detoxify these naturally occurring chemicals or even chemicals that have been added to the food, and they can induce an innate immune cell reaction and all the sequelae without raising antibodies. It's a pharmacological reaction. These chemicals are actually too small to raise antibodies. So the concept here is that the LCAT test is looking at a final common pathway, and I'm convinced that's the reason why it correlates as well as it does with actually eating the food. So the conclusion they came to was that the induction of NETs containing pro-inflammatory mitochondrial DNA might provide the clinical link necessary for a better understanding of the pathogenesis of non-IgE mediated food allergy, which again is typically a delayed reaction and more difficult to diagnose because it is delayed, and symptoms are not as acute. That's a topic for another lecture, but of course we know that type 1 or classical allergy to a food, be it a crustacean, for example, where someone eats a crustacean, gets hives right away, or has even a small amount of the protein in peanuts, it can produce a very dramatic effect, even an anaphylactic reaction. That's not what we're looking at right now. We're looking at these more delayed, nuanced, subtle, chronic inflammatory conditions, which are a little bit difficult to tease apart. There are, as we mentioned, there's a number of studies coming out, and it's increasing each year, talking about neutrophil extracellular traps, NETs. And this was of interest to me because it summed up a couple of points that I think relate to today's topic. So the dark side of neutrophils, neutrophil extracellular traps. These folks are from Sweden and Copenhagen, from government laboratories. So NETs contain antigens, some of which are modified histones. Now histones are, the DNA in the nucleus is not just floating around, it's wrapped around these octamer proteins called histones. And these protein structures have a positive charge and DNA is negatively charged. So it keeps it tightly wound around there so that not all the genes are expressed, only when there's a modification to the histone is there a change in the electrical charge, and that DNA can then be accessed. But when this netosis process occurs, there's a decondensation of the chromatin, meaning the DNA and the histones. And then there's a merger of these contents with these mediators like elastase and myeloperoxidase and other chemicals and the membrane proteins forming these nets. And that's been shown to be induce formation of autoantibodies, because the histones and the DNA that are toxic will remodel the tissue, the target tissue, and then the immune system doesn't recognize it and produces antibodies. So this is a clear pathway to autoimmunity, which doesn't actually involve the specific immune system directly. So it can be acquired, but yet it looks like it's a genetic autoimmune condition. For example, although I don't have the slide here, regrettably, recently from Stanford, there's a group looking at the increase in diabetes. And they found that the group that does not have the genetic predisposition for developing type 1 diabetes are showing the greatest increase in the incidence of diabetes. So there's some other pathway besides the specific immune system. Although the two work hand in hand, it's very nuanced, but essentially the NETs and the neoantigens that are created when tissue is modified by these toxic histones can create autoantibodies. The NETs were discovered as extracellular strands of decondensed DNA in complex with histone and granule proteins expelled from dying cells to ensnare and kill microbes. But they may spur autoantibodies and serve as scaffolds for thrombosis, providing a link amongst infection, autoimmunity, and thrombosis. So in this graphic, this is what the histones essentially look like. The DNA is wrapped around there. There's an enzyme, peptidyl arginine deaminase, which will change the histone charge. It will citrullonate the tail of the histone. It becomes a negative charge. It decomposes, and then we get then the possibility for the decondensation of the nuclear material. And when that happens, as we see here, we have a neutrophil forming a net. It can go down and hit different tissue. Arthritis, we have anti-nuclear cytosolic antibodies being created against citrullinated histones and causing arthritis, causing myeloperoxidase, causing vasculitis. And I think of interest today with what we're facing in the pandemic and blood clotting induced by innate immune cell activation is this pathway of neutrophils, and which again induce citrullonated histones, causing and DNA in the elastase, which is a major factor that ALCAT identifies the cause of, forms a scaffold for activated platelets, and we get thrombosis. And I think this is a key factor in terms of what we're looking at right now. There's a number of papers, this is just one, showing that COVID inflammatory microvascular thrombi are present in the lung, kidney, and heart of COVID fatalities, and they contain extracellular traps associated with platelets and fibrin. One of the other things, this is a recently published paper in British Journal of Gastroenterology having to do with, again, a totally separate ALCAT investigation from the University of Texas, looking at metabolic health, obesity. And this was a longitudinal case control study looking over the course of 1 year, putting people into one of 4 different groups, either an ALCAT avoidance diet together with aerobic surge exercising, ALCAT diet alone, exercise alone, and neither. And looking at outcomes in terms of waist measurement, BMI, and scale weight. And a good drop amongst those people who were doing exercise in both weight, BMI, and waist circumference. But even the group that didn't do any exercise, but just avoided their trigger foods, experienced nearly the same benefit, although I would never advocate for just changing diet. I think exercise, and I'm sure everybody would agree, in the right proportion for each person is about the healthiest thing you can do. But if you had to choose food allergy, which you don't, you can do both, food allergen elimination produces a more profound effect than exercise alone. I wanted to just bring this up a little bit because we've been talking about the innate immune cells. And this is actually an immune cell, a certain kind of dendritic cell that talks to a T cell. And these are specific cells. Now, they're called specific because T cells and B cells are specific to the pathogen. So not all T cells get activated when there's a certain pathogen. They interact, the process begins when a pathogen or an inciting danger molecule or an allergen is taken up by an antigen presenting cell, migrates to lysosomes where its peptides are broken down, where it binds with an MHC molecule, major histocompatibility molecule complex. So you have peptides with this MHC molecule. Now these are genetically determined, which is why certain people can eat gluten, certain people cannot, and the people who cannot, it's because of this molecule right here. It's the genes that encode for this molecule that enable it to bind with the gluten peptides and then transport it to the cell surface where it can activate a T cell, which recognizes those peptides in that molecule. If you don't have that molecule, you can't get celiac disease. That's why the genetic tests for celiac have such great negative predictability. And that's another group of tests that we do, genetic tests for celiac and Crohn's disease, and serological tests to see if there's active disease process going on. When the T cell gets activated, it then divides, because this, again, may be a memory cell, maybe a naive T cell that now comes to recognize this, or one that saw this before and is now remembering it. And then it multiplies, and the extent to which it multiplies is the extent to which it can directly fight infected cells or signal over to B cells that share the same kind of receptors to produce soluble forms of those receptors we call antibodies that correspond with the inciting pathogen or allergen. And depending upon which interleukin it is will determine the isotype of the antibody, which is very important because right now we know it's the IgG and IgM antibodies that show we have exposure. So they're good tests for determining whether we've had exposure, say, to COVID. Now, IgE, which is much less frequent, it's about 1/10,000th as frequent as an IgG antibody, will be the reagenic antibody, which leads to an allergy reaction. So that's a more complex system, it's more sophisticated, but they work hand in hand. And what's interesting is that amongst all the other diabolical things that NETs can do directly, they also prime T cells. So making their threshold for activation at a much lower level. So again, this can aggravate the Yang side that we really don't want to happen. So I guess at this point, you're getting the sense that our concept is amongst everything else you do, all the supplements you take, all the programs you do, all the stress management, you have to have a diet that's right for you, otherwise you're swimming upstream. And just to take it a little bit further, I want to mention, because as we saw before, that the specific immune system is responsible for producing specific antibodies to fight something if the innate immune system has not achieved resolution of that attack. The specific immune system, antibody affinity maturation will become more and more precise upon repeated exposure. Again, that's the concept underlying vaccination. You're exposed to something, the spike protein, whether it comes directly or we're induced to manufacture it. The specific immune system gets familiar with it, recognizes it more readily on re-exposure, and consistent re-exposure continues to modify the antibody to be more and more precisely have greater affinity with that. The innate immune cells also undergo changes, but not in their genes, but in their epigenetics. And this is a very cool paper that appeared in the New England Journal, showing how, in order, again, to maintain balance, the innate immune cells undergo epigenetic modifications, not genetic rearrangements like the B cells to produce antibodies, but changes on the epigenetic level. And therefore, it's really the epigenetic signature of the person that determines how they respond to future exposures. And one of the other interesting things is that the Western diet has been shown to cause epigenetic changes that train the innate immune system such that a secondary stimulus, in other words, the system is primed now. The secondary stimulus can produce a much more Yang type response and go overboard, and potentially it would be pathogenic. So watch out for that, which we all know. Western diet triggers NLRP3 dependent innate immune reprogramming. So all those other factors on the previous page from the recent New England Journal, we're talking about pathogens reprogramming the immune system. Now we're talking about how Western diet reprograms the immune system in a way which we don't want. And just in conclusion, I want to bring out one further concept that NETs are associated with. So once we have the activation of these cells and they die, we have to clean them up. Now, of course, there's actually, give or take, 100 billion cells that naturally die every day, and they have to be removed. And the macrophages in the system, the monocytes in the blood, and the macrophages in the tissue will remove those and do so silently. In fact, when it removes cells that die of apoptosis without having been activated, they actually reprogram the macrophages towards an anti-inflammatory or resolution type phenotype, an M2 phenotype, which is the yin side of the immune system. And it causes upregulation of things like interleukin 10, which are anti-inflammatory. However, if the macrophages have to clean up a lot of this cellular debris from activated cells, again, we saw the pathways, these DNA molecules that are circulating will activate through toll-like receptors, macrophages, and make them pro-inflammatory. But the whole gestalt of this dead cell and all this paraphernalia that's with it will program the macrophages towards a pro-inflammatory stance, which will perpetuate this. And it's done through certain cell surface molecules that are generated on the surface of the macrophages. And they respond to, and believing this is a technical term, it's not meant to be cute, find me signals and eat me signals that are expressed on these dead and dying cells. And what happens is that nets, diabolical as they are, I mean, they're good when you need it, but undue activation of the innate immune system on a chronic basis will cause all these issues. And the last thing they do is, if insult wasn't enough, they have to then inhibit the ability of the cells to clear away this junk. So here we have a neutrophil going into mitosis. Here's a macrophage, which would be quite a bit bigger, which is trying to digest this and neutralize it. There's a molecule which is particularly involved. This again is one of these eat me signals that's mediated by a molecule, MFGEA, which stands for milk. God, what does it stand for? Milk something epithelial factor 8. It comes from milk. And it helps the macrophage through integrins on its surface bind to the apoptotic cell with the mediation of this opsinin, which binds to the apoptotic cell. But the nets will inhibit that. So all the way around, you can get a vicious cycle where the inflammation produces more inflammation, becomes chronic. And I think this is a foundation that is important for everything that we're looking at, all the kinds of diseases ranging from IBS up to cardiovascular diseases, metabolic disorders, which are all interlinked, and even the complications from the more severe COVID fatalities or reactions, including the fatalities. Thank you for that. I'm going to turn this over to Amy, who will explain what you practically do with this information.
Hi, everybody. It's great to be here. Thank you, everyone, for asking us to be here. And in the interest of time, I am just going to skip my slides. And I think you all have a copy, a PDF of all of our slides. So you'll see that this, last part of this presentation is just to present a case of a patient who experienced great benefit from the ALCAT test and identifying her reactive foods, and at the same time giving an idea of how to break down the test results and how we suggest they get explained to patients and point out some important things about that. But basically, this patient, who we'll call Cheryl, she was struggling with weight. She had GI issues. She had inflammation. She shared some conventional blood work with us. Her estimated average glucose was elevated. Her A1C was elevated above the optimal, and her C-reactive protein was also elevated. She had symptoms of constipation and gas and bloating, but her big issue really was the reason for coming to see us was to lose weight and to reduce inflammation. So it was because she really was not an unhealthy eater. Excuse me. And when she came to see us, she was following intermittent fasting. She was avoiding dairy already. She was eating plenty of fruits and vegetables and taking protein powdered shakes, but good products daily. She was following a personal trainer's suggestions for a few months. So she actually was what you would consider a person following a health promoting eating pattern, but things were still not working for her. So it was decided for her to have the ALCAT test done to identify what food culprits could be working against her weight loss efforts and contributing to inflammation and her GI symptoms. So these are actually her results. So she was tested back in June of last year. And one thing that we feel, I'm going to show you the whole page here. This is the first page, 250 foods here on this first page, 250 foods reported here. as Roger described, the ALCAT test is not a test for the IgE-mediated food allergy. So I think it's very important that we differentiate with patients the different adverse reactions to foods, because it's confusing to patients. So, and even before they get tested with the ALCAT test, it's important, I think, for you to let them know that this is not an allergy test. So if your patient has an allergy to, like, say, shellfish, doesn't matter what the ALCAT test says about shellfish, they need to avoid shellfish. This is not a test for the type of the IgE-mediated food allergies. Cheryl did not have any food allergies, by the way, although she had an allergy to hair dye. Then there are intolerances, and we're trained to think of intolerances, of course, as the lack or the insufficient availability of a particular enzyme that is necessary to digest a particular component in food. So we're all familiar with the lactose intolerance, fructose intolerance, FODMAP, and many people are coming to us, interested in having the ALCAT test done because they're following these or avoiding these things and following FODMAP, and they're not seeing the results that they expect to see. So it's important to explain the different adverse reactions and to say, this is not a blanket diet here. This is what your cells are reacting to. As Roger said, the inflammatory response is happening from the leukocytes. So this, again, her test results, I think it's important always to focus on the good news. So we, took her right away to the green box here and got an idea of what she enjoyed from the green box. I think that's, important to share the good news first. The degree of reactivity in the ALCAT test is designated with the color coding system here. So again, green is non-reactive, and then the severe would be in red, moderate in orange, and mild in yellow. The general recommendations would be to eliminate the severe reactions for 6 months or more, the moderate reactions for 3 months or more, and the mild list, the general recommendation would be to limit them to 1 day every 4 days or 2 days in every 8. Now, I say general because obviously it's important to individualize for our patients. So we do recommend going through this with your patient in detail, or allow us to do that with you or with your patient. But what we did with Cheryl, and what I could tell you was significant for her in the severe list, believe it or not, she was not consuming apples, but beef a couple of days a week. Shallots, I was very surprised at. She was having a lot of shallots. She just loved shallots, so she was using that a lot in her recipes. And the protein powder that she was having daily had sunflower in it. So, if you share these results with your patient and you find out that severe list, you go through them and the patient says, yeah, I really don't eat those things. So you figure, okay, they're already avoiding those and they're still symptomatic. So it might make you want to tighten up the rules a little bit with the mild list. So this is why it's important to individualize. The moderate list, what was significant for Cheryl was anchovy. She was having Caesar dressing a couple of days a week. Often when I see anchovy or sardine or both, I think of fish oil. So it's important to be aware of that. It's not just about the food, it's about supplements, it's about personal care products. And Cheryl was not consuming fish oil, but as you know, anchovy and sardine are very common fish oil sources in the good fish oil products. But so just to make you aware of that jumps out at me. Avocado, she was having guacamole. She was consuming eggs a few days a week. She was having rice daily in her protein powder. Walnuts she was having, and potatoes. So those were the most significant ones. And what I want to point out here is a lot of times patients will say, well, I thought salmon was, one of the healthiest fish to consume. I thought walnuts were anti-inflammatory. Broccoli, I thought it was good for me. And the point is, yes, they are in and of themselves health-promoting foods. They are considered anti-inflammatory, but not for her right now. They are actually pro-inflammatory. As Roger showed you, the graphic of the cells changing in the number and the size, releasing those mediators, inflammatory substances into the circulation. So when you put it in perspective like that for a patient, they realize, oh, okay, no wonder I've got inflammation. So the other thing that very often happens, you'll hear from patients, they might look at their severe list and say, oh, come on, beef, really? I eat beef all the time and I don't have a problem with beef. And my response is, Well, yeah, you do. That's the point. You're looking for symptoms that would happen immediately after ingesting the offender, like would happen in type 1 IgA mediated allergies, minutes after ingesting the offender. That's not how it is with the food sensitivity response. The symptoms are more response that they'll experience with food sensitivities tend to be more chronic and more vague. So when you put it in perspective like that to a patient, it's, all right, even though they might not feel that inflammatory response, inflammation is happening whether they realize it or not. Then at the bottom of the page in the ALCAT test, they're looking at Candida albicans, gluten, gliadin, casein in the whey. The reason why these test components get their own box is because when they're reactive, it would require the elimination of more than one food. So they get their own boxes. So we all know what Candida albicans is, a yeasty-like fungus. When reactive in the ALCAT test, it's not a diagnosis for Candida overgrowth, but it does make us suspect that the patient has it. So we need to take steps to avoid the sugar. We would recommend antimicrobials, but we only recommend things that were tested and non-reactive. So you might want to suggest caprylic acid, which is coming from coconut. We can't suggest that because she was reactive to it. You might want to suggest garlic. Well, garlic was a mild reaction. Well, you can't suggest that very much. We could suggest oil of oregano. If a person is tested for golden seal with berberine, we can suggest that if non-reactive. So my point is only utilize things that were tested and non-reactive. Gluten, gliadin. She didn't react to gluten, but she reacted mildly to gliadin. So this was significant for Cheryl. She needed to be sugar-free. She needed to be gluten-free. She was already dairy-free. Because I thought her symptoms and her results were pretty significant with meaning she was consuming these prior to the test. I knew this was going to make a difference for her. We kept the mild list in 2 days per week. So ultimately, you're following the 5R program. We all know 5R, remove. The ALCAT test is telling us what to remove. The ALCAT test is telling us what foods to replace. We replace digestive enzymes if need be. Re-inoculate. We suggested Saccharomyces boulardii for Cheryl. And repairing the gut healing nutrients, of course. So the goal is that your patient, after following these results, following your 5R program, they've given the immune system a rest from the reactive foods. They've healed the gut. And you want to hear the patient say, oh my gosh, I feel wonderful. I feel, for all the reasons that what prompted the test, everything is being resolved, or it's already resolved. I feel wonderful. My weight is responding. My digestion is good, which is exactly what happened to Cheryl. You can see in her slides, 6 months later, body fat percentage came down. Her C-reactive protein came down. She's no longer constipated. She's sleeping an average of 7 hours a night, no longer has gas and bloating. If that's what you want to hear the patient say, then you'd be ready to reintroduce these previously reactive foods systematically, one at a time, to see if the symptoms return, because these are not necessarily forever. And that's another kind of confusing a question that patients have. Don't I need to avoid this for the rest of my life? No, your immune system is changing like every system in the human body. So what you're reactive to could change. And the goal would be that she's no longer reactive to all these things. We've healed the gut and we have encouraged a variety of foods. So she'd be able to tolerate these things again. The last slide you'll see in your slide in the handout is about Previmedica. We are the sister company to Cell Science Systems. We've been around for almost 10 years doing virtual nutrition therapy to people all over the country. How we help Cell Science Systems practitioners is we give complimentary review of all the test results that Cell Science Systems, all the tests that Cell Science Systems offers. You could always have a review with either myself or someone on my team. I have a wonderful team of nutritionists, or your patient can do that. We customize meal planning tools. You could participate in that service. We could make meal planning tools, which is exactly what we did for Cheryl. And we looked up special products that would work with her results. We do that sort of thing as well. We would provide the booklet for you with your practice logo on it to share with your patient. And we also, if you don't have a nutritionist on your team, we're happy to be the nutritionist for you. From where we are in Deerfield Beach, Florida, and I also have nutritionists on my network in other states too. So that's just a little bit about running through the ALCAT test results. I'm sorry, I just wanted to make sure that we had some time so that you could see what the test results look like. This is just the food page. There are many other test agents that the ALCAT test tests for. So, Tiffany, that does it for my part of the presentation. I think we're over time.
Great. Thank you so much, Amy. And thank you, Roger. We appreciate Cell Science sponsoring this lecture.